Hello, everyone. Thank you for joining us for this virtual demonstration of the ion torrent Genexus system. My name is Andrew Hutchison. Please enjoy this short demonstration using our new virtual demo environment, after which I will answer your questions. Throughout the presentation, please submit your questions to the chat, and I'll get to as many as possible after the demo. This is the integrated ion torrent Genexus system. This unique system is the only true end-to-end NGS system available today from a single provider. The system automates all the key steps of the NGS workflow. At a high level, these steps include sample prep, library prep, template prep, sequencing, and data analysis. Before we dive into the workflow, I want to point out the variety of sample types that can be run using the Genexus system. These sources of genetic material include tissue, blood, plasma, and several other sample types used in oncology and inherited disease research applications. The Genexus supports a variety of clinical and translational research applications, including genomic profiling for oncology and hemato-oncology research, and SARS-CoV-2 genome sequencing. Let's get started at the beginning of the specimen to report workflow and focus on the initial steps of the Genexus purification system first. While it is certainly an option to run these units in standalone mode, the integrated configuration provides the most seamless end-to-end workflow experience. So let's review this option today. The process begins with creation of a sample to result run plan with which is appropriate for the intended assay. As depicted here, to load samples, simply remove the foil seal from the purification plate and pipette your pre-processed samples into the wells indicated by the run plan. Users then load consumables onto the Genexus purification instrument. As shown here, proper consumable placement is detected in real time to prevent setup errors. If no errors are detected, the run can begin. At the end of the purification run, a 96-well plate is produced that is ready for transfer to the Genexus integrated sequencer. Users then move the plate to the sequencer. And then you load the remainder of the sequencing reagents onto the deck of the system. And again, real-time feedback helps prevent any setup errors from being made. The final step before the run begins automatically is to place the bulk bottles and sequencing cartridge into the proper spots and close the doors of the system. You can now walk away. It's really that simple. At the end of the run, the instrument automatically decontaminates with UV lights and the onboard cleaning solutions. Simply come back when the run is complete to view results. The Genexus software automatically executes base calling, alignments, and variant calling. You can view run metrics and QC reports to check your run and data integrity. And the only thing remaining is to generate a report through your lab's reporting solution, like Oncomine Reporter. That's really all there is to it. The Genexus system is truly an end-to-end solution. You can generate next day results because of the highly automated nature of this system, with only 20 minutes total hands-on time. It's been my pleasure to showcase the Genexus purification system and the Genexus integrated sequencer for you today. If you are interested in trying a virtual demonstration yourself, this experience is available in the Genexus System virtual lab. Now, I'll be happy to answer any of your questions. Um, and if you didn't catch it there on the last slide, we did want to call out that that virtual environment now has support for multiple languages, not just English. So if you're looking for and seeing if your language is actually supported, please do check it out and see if the language that you prefer is on there. So let's start with a few questions. We have a few preloaded from before, so we'll start with these. So there have been a few recent sequencing instrument launched recently, including the Element Biosciences Aviti, the Singular Genomics G4, the Illumina NovaSeq X, just to name a few. And how does the Genexus compare to these? So it's right. It's really an exciting time in NGS right now. All these instruments have advantages in different applications, and the competition and technology advancements are great for the future of basic science and translational research. The Genexus stands apart from these other systems on the market in that it was designed to be extremely fast and easy to use. It's still the only NGS platform available that offers a true end-to-end specimen to report walkaway automation from one single vendor. The Genexus is well suited for operators of all experience levels and is perfect for many clinical research applications, such as precision oncology, reproductive health, infectious disease, and inherited disease research. Next question. What is the cost to sequence a whole human genome on the Genexus? So we generally don't sequence whole human genomes on the Genexus. That's just not what it was built for. Many NGS instruments are developed for applications that require more sequence coverage but lower sequencing depth, like whole genome sequencing. On the other hand, the Genexus was designed for targeted NGS in clinical research, investigating smaller numbers of genes or genomic regions associated with various cancers or other diseases. In many cases, targeted NGS is the more economical, efficient, and effective approach, but in cases where whole genome sequencing is required, other instruments that have higher throughput and less automation would be appropriate. See a question, how do we calculate the DNA concentration after extraction without purification systems? So it is important to actually calculate the DNA concentration before you load it onto the Genexus sequencer if you are not using the Genexus purification system. That's because the concentration of your input going into the library prep reaction is important. You don't actually have to do the dilution, you just have to know what the concentration is. Typically, we will suggest using a qubit to do the concentration measurements and then put that into the Genexus software as you load up the samples. And then you can actually either allow the Genexus sequencer to do that dilution for you on the instrument, or you can actually do dilutions off instrument yourself to save the input and then go directly into the sequencer with the appropriate amount. All right, can I run libraries that I have already made for other ion-torn instruments on the Genexus? So yes, the Genexus does support running library to answer runs. You don't always have to create library from nucleic acid at the start. And so that means you can use libraries that are compatible with other ion-torn sequencers or the ion code barcodes. Do I need to dilute my sample before placing on the Genexus? So just as we saw in the demo before, the Genexus purification system, if you're running it in integrated mode, does away with having to do any dilution. It actually transfers the measurement information to the Genexus sequencer, which can automatically dilute the samples for you. If you do decide to do manual sample prep or any other automated extraction with a different instrument, then you do need to actually do the uh, concentration measurements and provide that to the Genexus sequencer. Right. Can I run multiple assays on the instrument at the same time? So the Genexus GX5 chip has 4 separate lanes, and you can actually load a different assay for each lane. But when you're running multiple assays on the instrument in the same run or at the same time, you do need to be choosing assays that have the same thermal cycling profiles for the library prep. So there are some conditions around running either just AmpliSeq assays or just AmpliSeq HD assays at the same time. All right, next question. How many samples can I sequence per run on the Genexus? So the Genexus currently has the capability to automate library preparation for up to 32 reactions at a time. Now, I say reactions because AmpliSeq is targeted sequencing, which requires sometimes multiple pools to be able to tile across genes when you're looking for full gene coverage or just to get to all the targets that you're looking for. And so depending on the assay that you're running, the number of pools that you are automating there, you just divide the number of pools from 32 to understand how many samples you can automate on the Genexus at a given time. For example, take the Oncomine Precision assay when running for FFPE. That is a two-pool assay. And so if the Genexus system can run 32 reactions per run, that means it can automate up to 16 samples running DNA and RNA at the same time. All right. And currently, which kinds of tests are performed on the Genexus? So the Genexus system supports several different precision oncology and infectious disease assays, with more coming soon. The Genexus system is currently compatible with custom AmpliSeq and custom AmpliSeq HD assays, which includes our white glove service or our AmpliSeq on-demand panels, and supports the design and development of panels and assays to fit any specific research needs. And let's see another question here. What read length does the Genexus system support? So the Genexus system can sequence up to 400 base pairs. So typically when we're designing amplicons for AmpliSeq, that we'll look at amplicons that don't rise too much higher than the 400 base pair read length insert size, more typically between 200 to 400. How is the chip stored after usage if not all lanes were used? So because the chip has 4 lanes and you can access them, either 1, 2, or 3, or 4 at a time. If you don't use all the lanes on the chip is stored on the instrument, and it is actually ready to accept more sequencing within 2 weeks of the first initialization of that instrument. So once you start the first run, if you just use 1 lane, you have up to 2 weeks to complete the other 3 lanes of that chip before the chip will have expired, and we recommend actually cleaning the instrument and reinitializing before the next use. And the last question here, how many reagent strips do I need for my run? So this gets to the question about the consumables used on the Genexus system. The Genexus consumables are of a modular design in that you can actually mix and match the necessary consumables for however many samples you are multiplexing at a time, and also how many lanes of sequencing you need to run. So you don't always need to run 32 reactions or 16 samples. You don't always need to run 4 lanes on a chip. You can actually scale the sequencing down to suit the batch size and your needs at that moment. And so there are sets of strips that correspond to the amount of library that you want to generate, as well as sets of strips that correspond to the number of lanes that you want to sequence on. All of this is very easily demonstrated for you through the Genexus software with either a printout, or you can follow the on-screen GUI showing you exactly how many strips of each type you need to put on the instrument based on the run and assay type and samples that you have set up for that run to go. Right, I think that's the end of the questions. So with that, I'll thank you again for attending Oncomine World, and please do come and revisit us and check out the Genexus virtual demo. Thank you.